Bioluminescence, Unveiling the Genetics. Title: Bioluminescence, Unveiling the Genetics. Introduction. Bioluminescence, the ability of organisms to produce light, has fascinated scientists and nature enthusiasts for centuries. This incredible phenomenon can be found across various groups of organisms, including bacteria, fungi, marine invertebrates, and fish. Genetic mechanisms of bioluminescence. Bioluminescence is made possible through the interaction of several genetic components. The fundamental building blocks consist of a luciferin, the substrate that emits light when oxidized, and a luciferase, the enzyme responsible for catalyzing this reaction. In many bioluminescent organisms, the luciferin and luciferase are encoded by separate genes within their genomes. Variety in bioluminescent organisms. The diversity of bioluminescent organisms reflects the multitude of genetic mechanisms underlying this phenomenon. Some bacteria, such as Vibrio harvii, possess a single luciferase gene in their genome. In contrast, the firefly Photinus pyrrhus boasts an entire family of luciferase genes, each with slightly different properties, enabling different colors or intensities of light production. Furthermore, some organisms exhibit bioluminescence only at certain stages of their life cycle, indicating the presence of genetic regulators that control the expression of the genes responsible. Studying the genetics of bioluminescence. Understanding the genetic basis of bioluminescence involves a combination of molecular biology techniques and genetic analyses. Researchers use tools such as DNA sequencing and gene expression studies to identify the specific genes involved in the production of luciferin and luciferase. By comparing the genomes of bioluminescent and non-bioluminescent organisms, scientists can pinpoint the genetic differences responsible for this remarkable ability. This knowledge aids in unraveling the functional roles of different genes and provides insights into evolutionary patterns. Evolutionary significance of bioluminescence. The evolution of bioluminescence has clear advantages for organisms that possess this ability. In marine environments, many bioluminescent organisms use their glow as a defense mechanism, producing a sudden flash of light to startle potential predators. Some deep-sea fish employ bioluminescence to attract prey or mates, creating intricate light patterns to communicate their presence. Understanding the evolutionary origins of different bioluminescent adaptations sheds light on the intricate interplay between genetics and survival in diverse ecological niches. Biotechnological applications. The study of bioluminescence genetics has also paved the way for numerous biotechnological applications. The luciferase gene, for example, is widely used as a genetic reporter in molecular biology experiments. By fusing the luciferase gene to a target gene of interest, researchers can visualize and quantify gene expression levels. This technique has revolutionized the field of molecular biology, enabling the study of gene function and regulation in various organisms. Additionally, the development of synthetic bioluminescence pathways has opened up avenues for applications in biosensing, environmental monitoring, and medical diagnostics. Medical implications. Bioluminescence research has unveiled potential medical applications as well. Scientists have developed novel ways to harness bioluminescence for imaging purposes, allowing the visualization of biological processes within living organisms. For instance, researchers have engineered bioluminescent markers that can track tumor growth or monitor the progression of diseases. Such non-invasive imaging techniques hold great promise for early detection and monitoring of various medical conditions. Conservation and environmental impact studies. The genetics of bioluminescence also play a role in conservation efforts and environmental impact assessments. With the increasing threats to ecosystems, understanding the genetic makeup of bioluminescent organisms can aid in determining their vulnerability and designing effective conservation strategies. Furthermore, monitoring bioluminescent organisms can serve as an indicator of environmental health, helping scientists assess the impact of pollutants or changes in habitats. Unfortunately, it seems like you have reached the end of your conversation. If you have any more questions or need further assistance, feel free to ask. I apologize for the confusion. Here is the continuation of the article. Mechanisms of bioluminescence. Bioluminescence is a remarkable phenomenon that occurs in various organisms, including bacteria, fungi, marine species, and insects. The underlying mechanisms of bioluminescence differ depending on the organism. In bacteria, such as Vibrio fischeri, the genes responsible for bioluminescence are organized in operons. These operons contain genes encoding luciferase enzymes, which catalyze the light-producing reactions. Additionally, there are genes that regulate the expression of the luciferase genes and control the timing and intensity of the bioluminescent glow. Marine species, like the dinoflagellate Pyracystis fusiformis, utilize a different mechanism. They produce light through a chemical reaction involving a luciferin molecule, an enzyme called luciferase, and oxygen. The specific luciferin used can vary between species, leading to differences in the color and intensity of the emitted light. The genetics of bioluminescence. Unveiling the genetics behind bioluminescence has been an ongoing challenge for scientists. However, recent advancements in genetic techniques, such as gene sequencing and gene editing, have provided valuable insights into the genetic basis of this phenomenon. One key discovery in the field of bioluminescence genetics is the identification of the luciferase genes. These genes encode luciferase enzymes, which are essential for the light-producing reactions. Another area of interest is the regulation of bioluminescence genes. Researchers have identified various regulatory elements, such as promoters and enhancers, that control the expression of the luciferase genes. Understanding the intricate regulatory networks involved in bioluminescence gene expression is crucial for deciphering the genetic mechanisms that govern this fascinating trait.